it defines the different groups within the protozoa. So I might give you another example of true-false question. Which one of these microbes would have pseudopodium? And I might list flagellates, ciliates, amoeba, cyanobacteria, something like that. So knowing features of the microbes that allow you to distinguish the groups and separate them from the others will be useful. Same with fungi. Know the characteristics of the group so that if I gave you a definition, you would know, oh, that sounds like fungi. Taxonomically related might be a definition I would give you. I could say, which one of these groups of organisms are taxonomically related? Protozoa, algae, fungi, cyanobacteria. Well, cyanobacteria are, so I wouldn't know that in there. But you know what I mean? Something like that. Let's see, what else did we say about eukaryotes? Sure.
Related to algae, you said phytoplankton. What's the significance of phytoplankton?
Phytoplankton?
Yeah,
well, basically, they're going to be oxygen producers. Phytoplankton, you think about where they're going to be existing. They're going to be in this upper layer of any body of water, the upper zone, where you're going to have algae and cyanobacteria, anything that would be photosynthetic that is floating in the water qualifies as phytoplankton. Okay. Which separates it from zooplankton, which is anything floating in the water that's not photosynthetic. Well, of course, because this is where photosynthesis is going. So these guys are using the sunlight and the nutrients in the water to photosynthesize and make sugars. Those sugars then become the base for all life beyond that. So yeah, they are the basis to the food chain, for sure. Yes?
Coming back a tiny bit, you talked about the thermal electronic sector.
Mm-hmm.
Can you maybe go back over that one more time?
Okay. Without getting into too much detail, because then this is really chapter 6 information that we're getting into. But when I talk about the terminal electron acceptor, when we break down glucose or break down any kind of fuel molecule, any organism that breaks down a fuel molecule, at the end of breaking down that fuel molecule, the electron that they got from it that had all the energy, that energy's been spent. And so they need to get rid of that electron to continue the metabolic process. For us, oxygen serves as that electron acceptor. So when we've broken down glucose and we've taken all the energy out of that electron that we can, we then transfer it to oxygen and the process can continue. If we don't have oxygen available, then the process stops and we stop breaking down glucose. And this is what happens in our muscles when we start working out really hard and we're not getting oxygen delivered to our muscles like we should. And if you haven't been working out regularly and you go to the gym for the first time and you do that, your delivery system to your muscles probably isn't real good. And so what happens is instead of burning down the glucose completely and oxygen taking that electron away, we go into a fermentation step where we break down the glucose into pyruvate, but we've still got to do something with that electron. So we give it to pyruvate and turn it into lactic acid, and that builds up in our muscles, and then that gets carried away. But when you're sore the next day after a hard workout, it's because of the lactic acid that is built up in your muscles due to the fact that you went anaerobic during your workout, that you weren't getting oxygen to those muscles to carry that electron away. So organisms that are anaerobic respirers, they use something other than oxygen to accept that terminal electron acceptor. A lot of times it can be nitrate or sulfate or something like that. And those that ferment, they use pyruvate or some other organic molecule as that terminal electron acceptor. But it really, one of the ways to think of it, it's just the garbage man accepting that spent electron. We've got to get rid of it to keep the system going, and so that's why, in this case, oxygen for us, something that's anaerobic would use something different. Let's see. Last thing we said in this chapter on eukaryotic diversity, we talked about arthropods, how they served as vectors. We talked about two types of vectors, a biological vector and a mechanical vector. Be sure that you know the difference between. One of them requires a bite, and the other one is just a transport vehicle. And then we talked about viruses. So you should understand the general structure of a virus, the fact that it comes with a capsid and that it has genetic material. Some of them have an envelope. Understand that bacteriophage only infect bacteria, and then that those that infect animals only infect animals. We talked a little bit about the lifestyle in the cell. We have those that are productive. Once they've infected the cell, they just reproduce more viruses until they lyse the cell. Then we have those that are temperate, where they enter the cell and they establish a latent period, where they just sit there until they manifest themselves at some point later. We talked about this idea of transduction, a movement of genetic material, and how the virus serves to move that genetic material. We mentioned there's generalized transduction, where the virus can land or infect a cell and then integrate its DNA anywhere in the bacterial DNA. And then when it excises itself out, it takes a little piece of that bacterial DNA with it. And then there's specialized transduction, where the virus integrates itself in a specific location in the bacterial DNA. Then when it excises itself out, it takes a specific gene with it as well. We use specialized transduction to help us move genes around from one bacterium to another bacterium. Talked about animal viruses, mentioned the term zoonosis, which is a transfer of a virus from an animal to a human. We talked about, sorry?
That was zoonosis?
Zoonosis, yeah.
Z-O-O-N-O.
Yeah.
So the animal who gives us the virus?
Yeah, so the rabies is an example of a zoonotic disease.
What do you have to do to avoid that, are you?
Don't get bit, best thing, yeah. Also, we briefly talked about, when we were talking about the Spanish flu, this notion of antigenic drift and antigenic shift. So be sure that you're familiar with what those terms mean. An antigenic drift is the slow mutation of the flu virus from season to season. But an antigenic drift, or excuse me, an antigenic shift is going to be when you get two viruses infecting the same cell, and then those two viruses swap genetic material, creating a whole new virus. And examples of that, the more recent swine flu that we've encountered, the Spanish flu from 1918 was another example of that. It can be avian genes along with pig virus genes, along with human virus genes, all coming together to form new viruses. Yes?
So in a pathogenic or turn into a shift?
If you had, well, we wouldn't think of it that way. You know, if you've got slow changes, you can have, all viruses would be undergoing slow changes. So yes, even those that infect a common cell and undergo antigenic shift, prior to them doing that, they've been undergoing antigenic drift as well. So yeah, the drift is happening all the time. The shift happens when they're infecting the same cell and exchange genetic material.
So is it kind of like after a while it kind of turns into
No, we wouldn't say that. Okay, it's not that mutations caused by drift will cause a shift. No, that's not the case. Mutations caused by drift are just drifting. So the organism is just slowly changing over time. Shift is when it's an abrupt change that takes place because of an exchange of genetic material between two different viruses. And so it's an abrupt change that no one in the population is going to be immune to. Yeah.
So then, that kind of segue into my question here is the immunity. So in a shift, when it's a slow mutations, people have immunities. They have residual immunity, but when you have, drift, that's drift, slow mutations.
Yeah.
But when you have shift where two viruses create a new virus with exchange, do they, does anybody have any antibodies to the, do they pass on some of the antibodies, like antibodies in human beings that have been previously exposed to either one of the two viruses, or maybe both of the two viruses that created a new virus? So the person's already been exposed, now there's a new virus, and they have antibodies to both of the old viruses. Do they carry on an antibody to the new virus?
Okay, so if I understand what you're saying here, let's imagine that this is our virus here, and it has on it, studded on the surface, the different proteins, metabiglutinins and neuraminidase. Okay, so those are there. When antigenic drift is taking place, okay, so back to your immunity thing. So we've been exposed to this microbe, and we have antibodies that recognize this, and we have antibodies that recognize this. And so if we're exposed to it again, we fight it off, no big deal. Antigenic drift says that over time, next season, this small change might take place. Okay, so it's a small change on the surface protein, and most of us still have an immunity to it because we're going to recognize this molecule, and maybe we'll still recognize this side here. So we still have antibodies that would recognize it. The next year, we look at it, and there's been another change, but now this change takes place here. We're no longer recognizing this molecule at all, but we still recognize this, so we still have some immunity